and this is part of my work that I've done for my honors thesis this semester. And I'm going to be talking to you about modeling the spread of a wheat pathogen in the United States. So where did we get this crazy, funky concept, right? Well, it all started with this thing called stem rust, which is, you can see a picture of it right here. It obviously gets its name because it has this orangey type of rust, really, look, on the wheat. And it is actually a fungal pathogen. Its scientific fancy name is this, and I'm not a biologist, or else I'd try to pronounce it for you. But wheat rust has always been a problem, and in areas that it does hit, it usually destroys about 15 to 20% of the wheat crop there. It's, like I said, it's always been a problem. It was actually a big problem in the 1920s in the United States. There was a huge epidemic, and there's certain things that were done after that epidemic, and even before that epidemic, that kind of to curtail the wheat pathogen of wheat rust. So there's certain chemical, cultural, and biological controls. And what I mean by chemical control is basically a fungicide. So a farmer goes out and sprays their crop, and it gets rid of the wheat rust. A cultural control is something like a slash and burn tactic. So if we're trying to stop the spread of the wheat pathogen, if we kill all the wheat that is affected, it's going to stop it, right? So slash and burn type things for cultural controls. Then there's also biological controls, which are a little bit more fancy. And so if we take the 1920s wheat rust epidemic, for example, that was dealt with by inbreeding chromosomes, crossbreeding a rye plant with a wheat plant. And so once you started crossbreeding that, you started getting genes in that system that allowed wheat plants to be resistant to wheat rust. And then in the 1970s, they started introducing what's known as SR31 into wheat cultivars. So when we have different varieties of wheat, we call them a wheat cultivar. It's kind of the fancy name for it. And basically, so in the 70s, they introduced this SR31, which was doing wonders for the wheat community, the agricultural community, until 1999. There you go. So what happened in 1999? Well, a special variety of wheat rust was discovered in Uganda, and it spread throughout Eastern Africa, and it was resistant to this SR31 gene. So you can kind of see it started off in 1999 right here, and then you have this up to 2009, and then it kind of spreads up. These highlighted areas are wheat productions, and then the red dots are actually where UG99 was discovered. So this really sparked our research question. We really wanted to know, well, what happens if UG99 type wheat rust comes to the United States? Are we all, like Dr. Ross said, going to die? So I'm just going to talk briefly about, oh, well, here we go. So this is actually the acreage map for wheat rust, or for wheat in the United States. You can see there's a lot of wheat being produced up in the central northern plain region. And then this, the USDA is well known for obviously they're not ignorant of UG99. And so they have this wonderful map right here. And this is their estimated crop loss based on the 1920 data. So if we took what happened in 1920 and apply it directly to what's happening today, this is the type of wheat crop loss that we can expect. Dr. Ross and I decided that that wasn't quite good enough, and that we wanted to actually model, well, what happens, what changes if we actually can get a little bit more flexibility and if we can actually model it as it goes. So the first thing I want to talk to you today about is a typical SEI epidemiological model. So us mathematicians, we usually do what's called compartmental models for epidemiology. And so it's basically, we're going to break up the whole population, in our case all the wheat plants, into 4 separate classes. There's going to be a susceptible class, an exposed class, an infectious class, and a removed class. So susceptibles are just healthy people, healthy wheat plants. Exposed is people that have been exposed to disease, or plants that have been exposed to disease. So crops that have, wheat rust has landed on the crops, in our case. And then infectious are people that the wheat rust has actually infected and is actually acting upon. And then removed, in a general epidemiological model, it can mean 2 things. It can either mean that you have granted immunity to the disease, so things like chickenpox. When you have chickenpox, you don't usually get it again, so we would move you to the removed class. Or we can also, in our case, it's going to be whether or not a wheat plant has died as a result of the disease. So those generate this set of differential equations, which are very fancy, and some of you might understand them, but I'm not going to spend much time talking about them. And instead, I'm going to show you a picture. So this is the type of thing that we can see with an SEIR epidemiological model. And you'll see that we're killing a lot of wheat at this point in time. This light blue line is the removed class, and then this dark blue blue line is the susceptible, and then we have our infectious and latent down here. And so basically what this is showing is that wheat rust has become epidemic within the population, and actually we're only harvesting about 20% of our wheat rust, of our wheat crop, which is not so great really. But you can see higher numbers of loss with UG99, so greater than that 15 to 20% that I mentioned earlier. So, what did Dr. Ross and I did? We did this much fancier version, right? So this is equivalent to that linear thing that I showed you a couple minutes ago. And so if you start off as a susceptible wheat, and we're going to look at just a single region for the first case. If you start off as a susceptible wheat, if you start introducing wheat rust, then you can move to this latent period. So once again, people that are infected but haven't actually shown signs or symptoms yet, or wheat plants, I'm sorry, I always say people because epidemiology is usually associated with people, but we're using it to model wheat this time. And then so once the wheat rust starts to germinate, you move to these infectious classes. And we're actually going to model 3 separate infectious classes. And the reason for that is because as you get greater infectivity, you're going to produce more and more wheat rust. So as you have more infection, as you're more infected, then you're more liable to pass it on to other people. So the rate at which infectious level 3 infects other wheat is greater than level 1 infection. So as we intensify infection, and we're going to say once you get to infection level 3, that's when you want to start doing something. That's when the farmer goes out and goes, well, you know, oh damn, I have a lot of wheat rust in my crop and I need to do something. So that's when we're going to start doing fungal pathogen treatment and that sort of thing. Slash and burn, that's when we're going to start talking about that sort of stuff. And if we have a successful treatment, then yay, we've moved our wheat rust, our wheat plant that were dying to the susceptible class. We've completely cured them. If we haven't cured them, then they're either going to stay in the class with unsuccessful treatment, or they're going to move to the removed class. So if we decide to slash and burn, then we're going to also move them to the removed class, or if they die from the death of the disease. So this is wonderful, right? But you can't plug this into a model. You can't plug pictures in the model. So we need a set of differential equations, right? So this is our set of differential equations. And I just want to point out a couple of things with this, since this is actually what we've been using to model a single region. The first of which is moving from the susceptible to the latent period. So wheat rust for landing on the plant. And that's modeled by these parts of the equations. We also have moving from latent to the first level of infectivity. Moving from the first level of infectivity to the second level of infectivity. And moving to the second level to the third level of infectivity. And finally, we're going to start killing some wheat with moving from the third level of infectivity to the removed class. And then we're going to do what we want to do, which is actually moving things from the infected classes to the susceptible classes. So we're actually going to keep and actually cure the plant this time, which is probably a good thing, right? You can see these parameters down here. If you want, you can kind of read them. For most non-math people, they're not very interesting, so I'm going to skip them. All right, so what are we looking at? This is the sort of graph that we get. And so once again, you can see that the susceptible class is moving downward, which you'd expect. We want things to get infected if we're going to start infecting things, right? And we're going to start killing things. This brown line is the removed class, and then you have all of the 3 levels of infectivity, and the latent period kind of lower than that. And so at this point in time, we're killing about 40% of the wheat, which should be a little bit more realistic with UG99, or UG99 type wheat rust. So this is for a single region, right? But we want to talk about the United States. If we're going to model the whole section of the United States as just a single region, the parameters for that would be nigh impossible to figure out. And so Dr. Ross and I decided, well, what happens if we start breaking things up? So we're going to look at a specific strip down in the United States. And so we're going to kind of take it as a general case, right? And we're going to divide it up into 10 different cells in a strip of cells. And in each one of these cells, we're going to model it as if it were a single region itself. So we're kind of getting more fine-tuned accuracy, right? And so what do we see? Oh, and sorry. The other thing about this is that we need to do, we have the interactions within the region, and we just talked about those, but we're also going to allow for interactions within between the regions. So wheat rust in Texas can infect wheat rust in Oklahoma, because unfortunately UG99 doesn't care about state borders. So what sort of activity are we going to find with the interaction coefficients? And so this is how we're going to define our distance-based interaction. Farther, you are out, basically what happens, the farther you are out away from the current plot of land, the less likely you are to get infected by wheat rust. Makes perfect sense, right? And then we're also going to define this wind-based interaction, and that's going to be based on where the two plots are we're talking about, and that's this theta location, which is the angle of the location between the two, and then also the direction at which the wind is blowing. So if you're blowing northward, then you're not going, if all the wind is always blowing northward, you're not going to infect any wheat that's lower than you, any wheat that's southern than you, or south of you. And that's exactly what we decided to take for our first case. So in our situation, we're going to blow all the wind north, and we're going to allow for that infectivity to start occurring. And what sort of things do we get? Well, this is the type of stuff that we see. So this is the number of wheat plants, right? And then the node number, which you can think of the node as those cells. So Texas is going to be down towards the bottom. We're going to label that as the last node, and then North Dakota is going to be the first node. And this is the sort of numbers that you're seeing. And the reason that you're getting this awkward curve is because of the way that we have the wind blowing and the different interactions of the curve, the different interactions between the things. So if you're blowing all the wheat rust into one location, you're going to see less there. But you also have to have time for it to develop there as well, which is why you kind of get that bend in the curve. So what do we want to do after this? There's a lot of stuff actually that we want to keep going on. Hopefully we'll get time to it for over the summer. The first thing we want to do is fine-tune those interaction coefficients, both as a method of the distance and of the wind effects. And the reason that we want to do that is because it'll give us a much more accurate model. And then we also want to start incorporating the United States data, very specifically the geographical organization of the United States. We're going to break it up by county. And then also the average wheat production within those counties. Obviously, if you have more wheat here, then you're going to have greater numbers of infection than if you have right here. In our previous model that I just talked about, we were assuming constant wheat production throughout the whole region. And then finally, we want to incorporate certain climate patterns. So what's the average wind speed? So how far can we blow things? How far can that wheat rust spore blow? And also, what directions it's going to go. Certain things always blow east, certain things always blow north, et cetera, right? And then we also want to explore possible prescriptive measures. So what happens if we start slashing and burning? What happens if we start applying fungicide? What happens if we introduce a UG99 resistant wheat cultivar? And so that ends my presentation. I just want to thank Dr. Ross, the EMU Honors College, and also Drs. Lindsay, Casper, and Storm, who were instrumental in all of the review work. And if you're interested, you can read up on wheat rust. Thank you guys so much.